This chapter is from Chapter 4, Cell Structure. This chapter will talk about cells and the different parts that make up the cell. We'll build upon our knowledge of our previous chapters with the chemistry of life and the molecules of life as well. And we'll also talk about transport of materials across cell membranes so that our bodies and our cells can get everything that it needs to be able to function. Looking at cell types, some of the cells that we would be most familiar with would be animal cells and plant cells. So in this slide we see pictures of each, with the animal cell over on the left and the plant cell over on the right. One of the main things you'll notice right away that stands out in the plant cell is this green area here. Plant cells and other producer cells have chlorophyll, which is contained in these structures called chloroplasts, as well as a large area called a vacuole to store sugar. It really sets off this type of cell right away. Up in the corner here we have another type of cell that in size comparison much smaller than the other two and lacking a nucleus. That's called a prokaryote cell. Bacterial cells are prokaryote cells. The plant and animal cells each have at least one nucleus. Therefore they're called eukaryote cells. Here's a size comparison. The bacterial or prokaryote cell here is quite small, 1 to 10 microns. And up against a eukaryote cell, much smaller. Eukaryote cell 10 to 100 microns. We study cells because, well, that's what we're made out of. And it's the basic unit of life. In order to be called alive, you need to be made of at least one cell. All living things are made of cells. And cells as a functional unit make up our tissues, which then those make up our organs, and those make up our bodies. And when you really look at it, cells are the little workhorses in our body, getting many jobs done. In order to live, cells need to be able to do certain jobs. For instance, they need to breathe. They need to bring in oxygen and release carbon dioxide. They need to eat, meaning they need to take in nutrients and be able to make ATP, which is our source of energy, an energy molecule. They need to build molecules, especially the molecules of life that we've talked about, the proteins, carbs, fats, and nucleic acids. They need to get rid of wastes. They need to regulate their internal environment, which is called homeostasis. It means a dynamic balance. They need to respond to their external environment, sometimes allowing things in, sometimes letting things out. And they need to be able to make more of themselves for growth and repair, for reproduction, and for development. Their three main jobs, though, are indeed making energy, making ATP. They make proteins, and again, proteins are the workhorses of the cells inside and do so much for our bodies. Making proteins is so important in fact that the code to make the right proteins for your body lies within your DNA. And they need to reproduce. When things go wrong, parts get damaged, those parts need to be replaced. Organelles, which means little organ, are the structures within cells that have specific jobs that they do in order to keep the cell alive and therefore keep you alive. Think of it like a mini organ. Cells need power. They need fuel to be able to do the daily things that they do. Our energy currency in our body is ATP. Basically, it boils down to us taking in the nutrients that we need every day, and your cells taking those nutrients, especially in the form of glucose, and turning that in to ATP. Unfortunately, doing that, wastes do build up like carbon dioxide, but those can be removed by respiration. Organelles that do this work, well, the cell membrane, the lysosomes, the vacuoles and vesicles, and the mitochondria, and we'll talk about each of those separately. The cell membrane helps to separate what's outside from what's inside. It acts as a gate or a barrier. It controls what enters and leaves the cell. Cells need things like oxygen and carbon dioxide, food and water, and they need to get rid of waste. So cells need to be what's called semi-permeable, meaning they allow some things in and some things out. That's also called, besides semi-permeable, it's also called being selectively permeable. They also need to recognize symbols or signals from other cells, and that's called cell communication. These protein molecules on the cell membrane here, they're shaped in a certain way to receive certain molecules. Up here, we talked about these in our last chapter, the phospholipids. They're a type of lipid that has a phosphate on the head up here, which gives this area a negative charge. And being that it's negatively charged or polar, this area likes water. And the area on the inside here is not charged. It's hydrophobic. It does not like water. So these phospholipids aggregate like this in what's called a lipid bilayer. So when things do pass across cell membranes, they need to be dissolved across this lipid bilayer, that is if they're nonpolar, or they need to find their way through a protein molecule that will receive them. The cytoplasm, important part of the cell, is the jelly-like material as well as the organelles that lie here in the cell, from the nucleus onto the cell membrane in between here. The cell membrane is the boundary. It recognizes things that should be in and should be out of the cell. Vacuoles tend to be places where items can be stored, as well as vesicles. Vesicles are also good for transporting items within a cell. It's like a little package. For instance, this cell has just taken in a small food particle and has made a vesicle around it using its lipid bilayer. And we can fuse that with another cell organelle called a lysosome that has digestive enzymes and start digesting that food. Here's one over here that has fused with an old mitochondria part that's within a vesicle to digest that old cell part. We don't want those old cell parts just hanging around now, do we? And building up. We need to get rid of them. Plant cells have a nice big central vacuole for storage right here in the middle. And this little organism here, this is a protist called the paramecium. And a paramecium, who's in fresh water, tends to take on excess water. Water likes to move from where there's more water out here to where there's less, to where there's more concentration of solutes inside the paramecium. And therefore our poor paramecium could fill up and burst with water. But luckily it has these little vacuoles that collect the excess water and then contract to expel it. And over here in the animal cell, we have some, like we saw before, food vacuoles. And here they are represented in this drawing. Lysosomes are little organelles that contain digestive enzymes. They can digest the food or old cell parts. I like to think of them as the housekeepers of the cell. By the way, all of these little cell organelles are contained by phospholipids, by little membranes of their own, separating themselves from the cytoplasm of the cell. Here's an interesting job for lysosomes. Did you know that in our very beginnings, while we're developing, that our fingers and toes actually have tissue in between them? But luckily, lysosomes, releasing their enzymes, help to get rid of that tissue in between. If that is unsuccessful during our development, we're born with what's called webbed toes or webbed fingers, as we see here. Here's a lysosome in this picture. Again, think of it like the housekeeper or the custodian of the cell. The mitochondria. I like to think of the mitochondria as the powerhouse of the cell. Here, sugar, like glucose, is taken in, combined with oxygen. And from that, we get some waste products, but we also get the most very important ATP, adenosine triphosphate, which is our energy currency of our body. Mitochondria have a lot of membranes inside here, inner membranes. You can see them here in the electron micrograph. That's so that it can be much more efficient with making ATP, to have more room for enzymes that do this process here of this chemical equation of making ATP. These are both in plant and animal cells. Here it looks like kind of a little bean-shaped organelle. There's our mitochondria. You can have many of these in a cell, and they can actually divide and make more of themselves. If you should start an exercise regime, you can make more, therefore become more efficient at making ATP. Going back a slide here, besides animal and plant cells, they're also in other cells as well. All cells need ATP in order to function. Sometimes we forget about that when we think plant cells being able to do photosynthesis and make their own food. But that's just making this part of the equation here, the sugar. All living things need ATP. So they have to take their nutrients, their sugar, combine it with oxygen, and make their energy currency. Luckily, they also have the chloroplasts, the plant cells do, and other producers, in order to make their own sugar. That also takes sunlight and carbon dioxide. Then remember, they can store it in that central vacuole. They can take that sugar and make starch by combining the glucoses, and then they'll have their nice little store for making ATP as they need it. Or animals like us can come along and take advantage of it and eat those sugar stores by eating the plant. Here's the chloroplast in a plant cell, and here's the mitochondria. In an animal cell, only the mitochondria. Cells need workers, and those are the proteins. They run the daily life and help the growth of the cell, etc. Proteins, again, can come in many forms, from being structural to being enzymes to being signals like hormones and receptors. They can also be our antibodies that function for our immune system. Organelles that do this work, well, the nucleus, the ribosomes, the ER or endoplasmic reticulum, and the Golgi apparatus. One of the major jobs of cells, again, is to make proteins, because proteins are like the workhorses of our cells and our bodies, doing so many things. Because they're so important, DNA actually codes for proteins. It tells the cell how to make them correctly. And DNA is found in the nucleus. And because DNA codes for proteins, the nucleus, which houses it, is sometimes known as the control center of the cell. The nucleus has its own membrane with pores. It even has kind of a nucleus of its own, called a nucleolus. The nucleolus manufactures ribosomes, which are the organelles that are outside in the cytoplasm, many times found on the ER, where proteins are actually strung together by stringing together the individual amino acids. DNA, when it's found in its organized state, wrapped around proteins, is called chromosomes. Humans have 46 of those. There's a picture of the nucleus with the nucleolus and the chromosomes. Ribosomes, think of them as the protein-making factories. They're able to read the instructions that are sent to them to build proteins from the DNA. Ribosomes are shaped kind of like two big globs that are on top of one another, down here in the right-hand corner. And a lot of times will be sitting on the endoplasmic reticulum. Sometimes they're free-floating out in the cytoplasm. And they're pretty small, so you can see them out here in the drawing or on the ER. The ER, or endoplasmic reticulum, are a series of kind of like membranous tubes that go throughout the cytoplasm. Think of them like little canals that you could go through to kind of move your way through the cell, through the cytoplasm. And because the ribosomes often sit on the ER, the ribosomes are making proteins, well, proteins can be transported through there. And that actually helps complete the proteins after the ribosome builds them. If ribosomes are on the ER, they're called rough ER. If they're not, then they're called smooth ER. And here's the ER. Either working on proteins or making membranes. The Golgi apparatus is usually found after the ER, but looks quite similar in that it's a series of little kind of tunnels or tubes or canals that go through the cytoplasm, and very good at pinching off to make vesicles. Think of them like the exporters of the cell. Kind of finish up the shipment, they finish up proteins, and then ship them out. And here's the Golgi right here. And I'm going to play this little animation here. There's our cell, the cytoplasm here, the nucleus, and over this red up in the corner is the bloodstream. So there's our nucleus. Here's our DNA. It's double-stranded. And DNA is quite the big molecule. It's kind of stuck here in the nucleus, but yet it has the instructions in its code for the cell to make proteins. But the items that actually make the proteins, the ribosomes, are found out here in the cytoplasm. So we need another molecule called RNA, which is only single-stranded and makes a copy of just one side, one of these spirals here of the DNA. Only one side, one copy is needed. And the smaller RNA is able to fit through the nuclear pores and travel out here to the ER. These little dots forming here are the ribosomes, so this must be rough ER. And our RNA is going to travel down here and get locked in by a ribosome who's going to read that code and string together the correct amino acids. So here we have a polypeptide, or kind of an unfinished protein. It's got to go through a few more steps to be considered finished, and that will travel through the ER onto the Golgi apparatus. We got to form a vesicle around it first, and the Golgi will help finish this protein. There, that's looking better. And we'll form a vesicle around it, pinching off into a vesicle, and probably transport it here to the bloodstream, where it will go off to where it's needed. Alright, here's our plant cell once again, looking at all the parts that we just covered. Cells need to make more cells. They need to reproduce so that we can replace and repair things that have died or been damaged, or in the case of children, we need to grow. Well, in order to make more cells, the new cells will need a copy of the DNA. We'll need more organelles, more cytoplasm. So once a cell is well fed and has made a copy of its DNA, then it will be able to divide into what are called two daughter cells. Organelles that do this work are the nucleus and the centrioles. The centrioles are these rod-shaped cylinders here. They help to coordinate cell division by coordinating little protein structures called microtubules, which can grab a hold of chromosomes and pull the doubled copies apart so that we get an even distribution of DNA to the new daughter cells. Here's the centrioles right here. Here's our summary. Cells have three main jobs. They need to make energy in the form of ATP, which will also have to remove waste from doing that process. The making of ATP is called cell respiration. They'll need to make proteins, because remember, proteins do all the work of the cells. Well, maybe not all the work, but most of the work. And they need to make more cells, and that will contain making more DNA and then dividing the cells into two daughter cells, making sure that each new cell has a copy of the DNA, the copy of the cell's instructions to making proteins. Peroxisomes are other digestive enzyme sacs that can be found within cells, in both plant and animal cells. They break down fatty acids to sugars. They're easier to transport and use as a energy source that way. They also detoxify the cell by detoxifying alcohol and other poisons, and they can produce peroxide, H2O2. You can break that down from H2O2 to the more familiar H2O, or water. One theory, called endosymbiosis theory, suggests that mitochondria and the chloroplast, the place where photosynthesis occurs in plants, were actually once free-living bacteria that were engulfed by an ancestral eukaryotic cell and then came to live within the cell, and because of the benefits of mitochondria making ATP and chloroplasts making food, they were kept. An endosymbiont, as in endosymbiosis theory, is a cell that lives within another cell, and the other cell would be called the host. It becomes a partnership. It's like an evolutionary advantage to have them within you because of the supply of energy and the raw materials in the form of sugar. And yet the mitochondria and the chloroplasts receive protection by living within this eukaryotic cell. Mitochondria and chloroplasts can move within cells and they can reproduce on their own, and they're about the size of a bacteria, and they actually have their own DNA to make their own proteins, lending evidence to the endosymbiosis theory. This is a diagram with a kind of a timeline in how that would happen. Here our cell takes on a mitochondria and chloroplast and engulfs it, and then they stay within and don't get digested. Now we have a partnership where the mitochondria and chloroplasts get protection, and the cell as a whole has something that makes ATP and sugar. We talked briefly about cells making more cells. I wanted to show you this slide of the cytoskeleton. If you think of a cytoskeleton as like the scaffolding, like a building has scaffolding, the cytoskeleton is that way as well. Look at these down here in the lower right corner. These are the microtubules made of proteins that help to hold the structure of the cell, to maintain its shape and support it, and to anchor organelles as well. These microtubules come in different sizes, from microtubules to intermediate filaments to smaller microfilaments. They also are part of the function of how a cell moves. If a cell has cilia, which are something like, we saw the paramecium earlier, like little hairs that help propel the cell, or like one big hair, like a flagella, a whip-like structure that helps propel it. And again, these are proteins and can contain the actin-myosin proteins like our muscles have to contract in order to help with this motility. Here's a neat picture showing how organelles are anchored by microtubules. And are also part of the centrioles that help to control cell division. Here's a neat picture. Here's a cell. You can see the chromosomes darkened. You can see the microtubules, what's called the mitotic spindle, and that cell is about ready to divide and pull the chromosome pairs apart. Here I'd like to revisit the cell membranes. Remember, we have the lipid bilayer, the double layer of phospholipids with the polar ends pointing out and the nonpolar ends pointed in. Within the lipid bilayer, we have different types of proteins. Remember, our proteins are kind of like our workhorses of our cells. This is a channel protein, allowing things to come in and out of the cell like a channel. Can also be called transporter protein, Here we have one that's functioning as an enzyme by holding certain molecules in place so that reactions can happen faster. A receptor protein receiving certain molecules, so they're shaped in a certain way to receive the right ones. Here we have a cell surface identity marker. This area on here, known as an antigen, kind of marks it as a self. This is you. Recognize your own proteins. Cell adhesion, in order to adhere cells to one another. And also a place to attach the ever important cytoskeleton. Here's another neat picture of a cell membrane, zoomed out a little bit so you can see a little bit above and a little bit below the cell membrane. These phospholipids are quite flexible, slipping past one another, switching places. We've got some cholesterol molecules mixed in here. We've got our proteins doing certain jobs within the membrane. And we even have some sugars sticking up from the top. Because this is kind of like a fluid entity here, it's called like a fluid matrix. The lipid bilayer or the cell membranes really are a remarkable cell part. Cells often have to be linked together, sometimes to exchange materials, exchange signals, or to just be connected to one another. Those are called cell junctions. Tight junctions are membranes that are fused and form kind of a continuous seal between cells. Desmosomes, also called anchoring junctions, fasten cells together into strong sheets like rivets, and keratin proteins anchor them to the cytoplasm. Gap junctions, sometimes called communicating junctions, are most similar to plasmodesmata in plants, provide cytoplasmic channels between cells where things can be transported across those little areas to neighboring cells. The plasmodesmata in plant cells are open channels, just like the gap junctions. They extend across cell walls and connect the cytoplasm of adjoining cells. Substances like water, nutrients, and signaling molecules can flow quickly from cell to cell through those junctions. Lastly, I'd like to talk about cell theory. Cell theory is made up of the following statements: that every living organism consists of one or more cells. The cell is the structural and functional unit of all organisms. A cell is the smallest unit of life, individually alive, even as part of multicelled organisms as well. All living cells come from division of pre-existing cells. And finally, cells contain hereditary material, which they pass to their offspring during division. The cell theory was first articulated in 1839 by Schwann and Schleiden, and later revised, and remains an important foundation of modern biology. It's one of the most important theories that we have in biology. I'd like to remind you to also read the beginning of our chapter 4 in the food for thought section, where they give us kind of a nice little story about E. coli and how many bacteria live within us, and we're okay with that, but how there are some strains of E. coli that make people sick and actually can cause death. And of course, that would be the kind of bacteria you wouldn't want within your body. They let us know that in 2006, more than 200 people became ill and 3 died after eating fresh spinach grown in a field close to a cattle pasture where water contaminated with the manure and the E. coli must have been used on it. Kind of scary. But make sure you read that and read kind of the finishing message at the end of the chapter. That finishes that out. Be sure to do the self-quiz and the other questions at the end of the chapter as well, and read the summary to help sum it all up.